Thank you, Dr. Kimwright. Hello, everyone. I am Alan Pepper, a PhD student from the University of Georgia Plant Pathology Department, and I am co-advised by Dr. Lee Young and Dr. Timothy Brenneman. And today I will be talking about my research in calcite dissolving bacteria as a new form of calcium source. So first off, well, all or most of you will know that peanut is very economically important to the state of Georgia, bringing in 663 million in crop revenue, with 800,000 acres dedicated solely to peanut farming in this region here in the southern part of Georgia. Georgia is the #1 producer of peanuts in the United States. However, with that, it has the second highest cost for fungal and herbicide control at 60 million, and highest total percent loss at 25% compared to other commodity crops. So just a quick reminder of peanut physiology. Peanuts are known as a geocarpy plant, meaning they flower above ground and fruit below ground. So the way this works is that the flower is pollinated and then the peg or geniform forms and then moves below ground where the pod then begins to grow and form. The ovaries are located at the tip of the peg, and as this ovary swells and the pod forms, it begins to actively uptake nutrients. It's very unique in the fact that the pod or the fruiting body itself is what is actively uptaking these nutrients, and very little is actually transported from the roots. So one of these important nutrients is calcium. So as many of you know, calcium is critical for peanut pod formation, and this information has been well studied, has been known from the 1940s as well, with this early study that showed here with available calcium, you have proper pod formation, but without only the pegs form and no pods are actually able to form. So without calcium, you get embryo abortion or POPs, which is shown in this picture above. Termination of the pod expansion, reduction in yield, increased disease susceptibility, and lower overall seed quality. There are a lot of different factors that influence calcium uptake. Pod shape and pod characteristics are part of that. So the thicker the peanut hull, the less efficient they are at absorbing calcium. And the genetics or the cultivar also influence the rate at which they're able to uptake calcium. So the only way to alleviate this need for calcium is by applying gypsum or lime at relatively high amounts. Gypsum is a byproduct of coal energy, and recently there has been an increase in power plant or coal production shutdown. And so with that, it's expected for gypsum to increase in cost. And so with that, a new alternative is needed. And so our solution to the need for a new source of calcium is in the form of microbes. So there are a group of microbes that are able to induce calcium precipitation and solubilization through a process of biomineralization, meaning they're able to convert calcite, the mineral form or insoluble form of calcium, into the soluble form of calcium, which then could be uptaken by the peanut pod. So the question is, how can we use these bacteria to create enough available calcium for the peanut plants? So I have 3 primary objectives for this research: isolating and characterizing these CDBs, testing their efficacy in promoting pod growth and seed quality, and the development of a medium throughput system for studying pod growth in response to calcium availability. So the first step in this is isolating the CDB. So this was done in Black Shank Farms, Tifton, Georgia. Soil was collected and then through a series of dilutions and plating onto a low nutrient differentiating media that is amended with 1% calcite. So this creates an opaque plate. And then as the bacteria grows, a clear zone or halo is formed around the colony. And this indicates that it is able to dissolve calcite into calcium. So the next step after isolation was identifying what CDBs we had. So we collected 64 CDB soil isolates from Black Shank Farms, and then the DNA was extracted using the V4, V5, 16S RNA primers. and then classified to the genus level. They can only be classified to the genus level using 16S because it is not as specific enough to give the species. They were then aligned, and then through BLAST and NCBI's database, we're able to identify them. And then on the left here, so you can see these are the sequence. So we had 23 fully sequenced. 10 of them are distinctly different, meaning not from the same genus. And then right here, we have an example of the morphological differences. So you can see that they have quite different characteristics. Some are pigmented, different colors, and also they grow at very different rates. So our primary focus is on the Bacillus and Pennebacillus bacteria, as they are endospore forming and are shelf stable. And so the end goal of all of this is to produce a form of biofertilizer. And so we would want a shelf stable bacteria that we can use and provide to farmers. So the next step is determining the rate at which they are able to dissolve this calcite into calcium. And so I developed a 6-well plate system. So again, this has this calcite agar with the 1% mediated media, and then a 10 microliter droplet of the CDB at 10 to the 6 CFU per mil is then placed in the center of the well. And then the ratio, or the rate at which the halo it forms comparatively to the colony size, is known as the solubility index, or the rate at which it is able to dissolve calcite into calcium. And so some quick results from that. If you look here, you can see that 99A and 100A, which are Leliodia, are able to dissolve calcite at a very high rate. And so this green line here is a Pseudomonas that is unable to dissolve calcite. So this is a baseline. So with all this, it indicates that all these strains that we collected are able to dissolve calcite. However, all of them, not all of them grow at the same rate or at the same efficiency. So this is all to characterize and fully understand and basically choose the best CDB for our future research. So part of that is actual application onto the seeds, and we wanted to determine if these CDBs had any effect on seed germination. So this is a random block design within our greenhouse where the seeds were soaked for 30 minutes in the CDB suspension and then repeated 3 times. And so looking at the study control, which was just water in this case, compared to many of our strains, a lot of these strains, 18.2 and 100A, increased the germination rate significantly compared to the control. However, we need to repeat these again and fully understand this. Also, you can see our germination rate is relatively low. This could be due to the greenhouse growth conditions. However, with that, we still had an increase in actual germination due to the CDBs. So the next way we were looking into characterizing these is by doing a soil drench. So we had a CDB suspension, and then it is added into the Tifton soil, and then we wanted to see the amount of calcium increase using a soil calcium test. So looking at our top one here, 95A, which is a Cellulomonas, it had a 91% increase in the amount of available calcium in that soil sample. And so this continues on. So you can see that some have very significant increases, some are relatively small when adding directly into soil. So the next look was looking at the actual population dynamics within various soil and rotation types. So we took soil samples from different field types: vegetable, peanut, cotton, onion, and cotton. And then looking at the calcium in the soil, which is on the right here, and then the CDB ratio, which is the percent of CDBs compared to the total colony population. We noticed a inverse or negative correlation between the amount of calcium in the soil and the amount of CDBs. So meaning as the lower the calcium level, the higher the CDBs are present in the soil. And we are going to do further research into this unique phenomenon. So kind of switching up a little bit here is how we're actually going to apply and study these CDBs. So I developed the in-tube growth system, or ITG system. So the system has a 50 ml tube with a hole in it, and then you can fill it with various media, in this case soil. It has a gas exchange system, so this has a layer of protective mesh that prevents the soil from interfering, and then some air holes, and then it's covered in micropore tape to keep out bacteria and maintain the humidity. So the peanut peg, before it touches the potting soil, is inserted into this tube, and then here you can see the peanut is actually able to grow inside this tube, and this creates a controlled environment. And as I said before, the pod itself is uptaking the nutrients, so we can change the nutrients within these tubes and look at how it affects the seed quality. So some quick results from the ITG system, looking at a visual analysis of open soil, meaning these are normally grown peanut plants, in the ITG system. We can see no significant difference between these two. Also looking at the seeds themselves, there is no major visual difference. Looking more statistically at them, we looked at the fresh pod weight, the fresh seed weight, the dry pod, and the dry seed weight of these samples, and determine if the tube was having a negative impact on the actual growth and expansion of the pod. However, we don't see any, there is no significant difference between growing these seeds in open soil and within our tube, which is great for our future studies. And to be sure, this was in Georgia 06G, which is a relatively small runner type peanut. We chose to do it in New Mex Hyoleic 01, which is a Valencia type peanut that is relatively larger. And it also had no significant impact growing a larger size peanut within the tube. And so with this, we're able to use this system to monitor and add in our CDBs for future studies. So in the future, we plan to continue exploring this unique trend that we saw between the relationship between soil calcium level and the amount of CDBs present. So I have conducted a survey of 3 regions in Georgia: the upper coastal plain, the deep sands in the southwest, and then middle Tifton, Georgia. And we have, I have completed this survey, however, I have not finished the analysis. But we were going to look more into this trend and see how calcium impacts the CDB population. And then the next step is the actual application of the CDBs into the ITG system. So we'll create a low calcium media inside this, and then apply the CDBs and see how it affects the seed quality, if it improves it or decreases it. So in conclusion, the CDBs are present in Georgia fields. We have isolated 63 of them and fully identified 23 of them. The CDBs can alter the soil calcium level. The ITG system can grow a single peanut pod in controlled conditions. And with this, the overall goal is to develop a CDB biofertilizer and reduce the need to use external sources of calcium like gypsum, and then also increase the protection from soil-borne pathogens. And with that, I would like to acknowledge all of my lab members, our funding from UGA and the Georgia Peanut Commission, my co-advisors, Dr. Leon, Dr. Timothy Brenneman, and the county agents that helped me with my survey, and to all of the farmers that also allowed me to access their fields. Thank you.
All right, thank you, Alan. We have plenty of time for questions. I've got a, let's see, here's a question. You're saved from my question. Here's from Dr. Dan Anko. He says, for the germination test, did the seed have standard seed fungicide treatment prior to soaking with CDB and planting?
Yes, they did have a fungicide powder coating.
Okay. And my question, Alan, would be this: have you worked with any of the soil fertility specialists, or have you done any kind of cooperative work with those involved in making recommendations or working with the calcium in the soil yet?
No,
as far as peanuts.
Okay. No, as far as I know, we haven't done that.
I would imagine there's tremendous interest from those who making the recommendations currently on gypsum and calcium to collaborate with you. All right. One more question. All right. Actually, you got two from Dr. David Langston. Do CBDs have the potential to reduce the rate of gypsum? And what if a soil does not have enough calcium to convert to soluble form?
Right. So for the first question, our goal is for this to be an alternative or a supplemental source of calcium instead of using gypsum. So we hope it can be used in sort of a irrigation kind of overhead soil drench system, and kind of used in the alternate years between gypsum applications. So gypsum is usually not applied every year. So this would be kind of a supplement between those alternating years. And then for the second question, that is an excellent question. We have tested it in very low amounts of calcium or calcite abundant sand and other things, and it still seems to increase the amount of soluble calcium. However, we're testing that further. And it will definitely depend on the soil type, and that's why we're doing a larger survey of fields in Georgia.
Thanks, Alan. That's the time we've got. Thank you very much for participating and for that presentation.